Did you know that your brain began forming only 18 days after you were conceived? Before your heart fully beats, before your face appears, your brain is already building the blueprint of who you will become. Today, we uncover the hidden world inside the early embryo, how the human brain truly begins its journey. Welcome to Medley Valin, your channel for clear, research-based medical and biological knowledge. Today, we dive deep into embryology to understand how the human brain begins to develop, what scientific stages it goes through, and which molecular signals guide this entire process. Let's begin our journey into the earliest days of human life. Our story starts around day 18 of embryogenesis, a critical moment following a process called gastrulation. Along the bottom of the tiny embryo, a flat sheet of specialized tissue appears. This is the neural plate, the very mother of our entire nervous system. At this microscopic level, a symphony of molecular signals is already at play. According to research, signaling pathways known as BMP, WNT, and the FGF become active. These signals act like microscopic conductors, instructing a group of cells to set aside their other potential fates and commit to becoming neural tissue. It's the first step in constructing the most complex object in the known universe. Next, between days 18 and 26, a truly dramatic transformation takes place. The flat neural plate doesn't stay flat for long. It begins to fold inward, curling up at the edges, bending like you roll a piece of paper into a tuba. This incredible architectural feat is called neurulation. The tuba then zips itself closed, forming the foundational structure of the human brain and the spinal cord. The closure happens in a specific sequence, the front end, called the anterior neuropore, which will become the brain, closes around day ke-25. A couple of days later, the posterior end, destined to be the spinal cord, closes around day 27 or ke-28. Scientific research shows that if this closure process fails, it can lead to serious conditions known as neural tuba defects, such as anencephaly or spina bifida. This stage is a perfect example of biology's precision engineering. Once the neural tuba is fully closed, we enter week 4. The head region of the tuba begins to swell and expand, forming 3 distinct, bubble-like structures. These are the primary brain vesicles. They are named the prosencephalon, or future forebrain, the mesencephalon, or midbrain, and the rhombencephalon, which will become the hindbrain. Think of these 3 vesicles as the foundational building blocks from which all the intricate regions of the adult brain will later emerge. The formation and the separation of these vesicles aren't random. Embryology research has confirmed that specific genes, like sonic hedgehog, WNT, and DOTX2, act as master regulators, meticulously controlling this crucial division and setting the stage for the next wave of complexity. As we move into weeks 5 through 7, the brain's architecture becomes even more sophisticated. The 3 primary vesicles now divide further, creating 5 secondary vesicles. The prosencephalon splits into the telencephalon and the diencephalon. The mesencephalon remains as is. And the rhombencephalon divides into the metencephalon and the myelencephalon. This is the stage where the basic blueprint of the brain is truly laid down. The telencephalon will give rise to the cerebral cortex, the seat of our consciousness. The diencephalon will form the thalamus, our sensory relay station. The metencephalon develops into the cerebellum and the pons, crucial for balance and the coordination. And the myelencephalon becomes the medulla, which controls vital functions like breathing and the heart rate. This intricate patterning is guided by molecular gradients. Signals like sonic hedgehog define the bottom, or ventral, identity of the neural tuba, while BMP and the WNT signals establish the top, or dorsal, identity. This creates a coordinate system that tells every cell exactly where it is and what it should become. From week 8 onward, the embryo officially enters the fetal stage, and an explosive process known as neurogenesis begins. Deep within the walls of the brain vesicles, in an area called the ventricular zona, neural stem cells start dividing at an astonishing rate. We're talking about thousands upon thousands of new neurons being produced every single minute. But not all cells become neurons. A delicate balancing act, governed by a signaling pathway called notch, determines the fate of each cell. Notch signaling essentially helps cells decide whether to become a neuron and stop dividing, or to remain as a progenitor cell and continue producing more neurons. This rapid proliferation is what builds the sheer number of brain cells needed for a functioning nervous system. Now, having a massive number of neurons isn't enough. They need to be in the right place. This brings us to the incredible process of neuronal migration. After they are born, newly formed neurons embark on a journey to their final destinations. They are guided by specialized cells called radioglia, which act like ladders or scaffolding, stretching from the inner ventricular zona to the outer surface of the developing brain. Neurons climb these ladders to reach their designated layer. The human cortex, for example, develops in a fascinating inside-out pattern. The deepest layers form first, and the subsequent waves of neurons migrate past them to form the more superficial outer layers. A crucial protein called renin acts as a stop signal, telling the migrating neurons when they have reached their correct position. Research shows that mutations in the genes controlling this migration can lead to severe cortical malformations, such as lissencephaly, a condition where the brain lacks its normal folds and grooves. With neurons in their proper places, the next monumental task is to wire them all together. Neurons now extend long, slender projections called axons, which act like biological cables, seeking out their correct targets to form the earliest neural circuits. This process, known as axon guidance, is a marvel of molecular navigation. The growing tip of the axon, called the growth cone, is like a tiny explorer, sensing its environment for chemical cues. These cues, with names like netrin, SLET, semaphorenes, and the ephrins, act as attractants or repellents, guiding the axon along a precise path, telling it when to turn, when to move forward, and when to stop. This initial wiring forms the fundamental framework for everything our brain will ever do, from basic reflexes and sensory perception to our future thoughts, emotions, and memories. As the fetal period progresses, the connections between neurons, called synapses, begin to format an incredible patch in a process called synaptogenesis. This is where the real communication network of the brain is built. After about 20 weeks of gestation, this network becomes active enough that early, coordinated electrical brain activity can be detected using sensitive equipment. The developing cortex, thalamus, and the brainstem start exchanging signals, practicing their communication. These early, spontaneous patterns of activity are not random noise. They are crucial for refining neural circuits and laying the essential groundwork for future high-level functions like learning, attention, memory, and sensory processing. The brain is essentially testing and strengthening its connections long before birth. Understanding this intricate developmental timeline has profound clinical importance. Medical research has firmly established that a deficiency in folic acid is one of the major preventable causes of neural tube defects like spina bifida. This is why prenatal vitamins are so crucial. Additionally, a mother's health and the environment play a significant role. Conditions like maternal diabetes, exposure to alcohol, certain medications, and even some viral infections can negatively impact this delicate process of early brain development, leading to lifelong consequences. By understanding these embryological mechanisms, doctors and scientists can improve prenatal care, develop potential therapies for developmental disorders, and enhance our ability to detect problems early on, giving every child the best possible start in life. Human brain development is undeniably one of the most complex and beautiful processes in all of biology. To think that it all starts from a simple, microscopic neural plate at just 18 days, and evolves into the trillions of connections that make up a fully functioning brain at birth, is a scientifically orchestrated miracle. It's a testament to the power of molecular signals, genetic blueprints, and the precise timing. If you appreciate authentic, research-based medical content that breaks down complex science, be sure to follow Medlivalent. Thank you for joining us on this journey. Please remember, this content is for educational and informational purposes only and is not medical advice.